So it's my pleasure to be here today, and thank you all for coming. Most of you are probably familiar with PacBio, but we are a long-read sequencing company. We launched our first system almost 8 years ago now, and since then we've had a variety of hardware, software, and chemistry improvements that have increased our read length more than 100 times, and we've increased our throughput per SMRT cell more than 10,000 times since 8 years ago. And that leads us to where we are today, which is earlier this year we launched our latest sequencing platform. This is the Sequel II system, and it uses our newest generation SMRT cell, which is the SMRT cell 8M. The 8M indicating that this is 8 times higher throughput than the original Sequel system. Of course, this also means that we have higher throughput and reduces the project time and project cost. And this system is particularly optimized with read lengths and software for high-fidelity sequencing, or HiFi sequencing, which is our new highly accurate long-read sequencing. I'll be talking about that in more detail shortly. First, I'm going to show you performance. So this represents our average customer yields per SMRT cell over the past 3 years. So in blue is the original Sequel system, and you can see that over the past 6 months or so, we've been pretty consistent at about 15 gigabase per SMRT cell. The green bars represent average yield per SMRT cell on the Sequel II system. So the first couple months here is from our 5 early access customer sites. These are sites that are highly skilled in PacBio sequencing. You can see that they perform very, very well. Since May, that's when we started installing sites for our general commercial launch. And since then, we've had increasing throughput per SMRT cell. And just this month, our customers are averaging about 160 gigabase per SMRT cell. So that's actually more than 10 times higher than we were with the original Sequel system. Reliability is very good. So across all of our systems, our SMRT cell failure rate is between 1% and 2%. So excellent performance across the board. I had mentioned that one of the things that we're really excited about is the introduction of high fidelity or HiFi sequencing. This is highly accurate long-read sequencing. The way that this works is on the Sequel II system, we take advantage of the very long read lengths to actually sequence single genomic DNA molecules multiple times. So we target maybe 8 or 9 passes to get to that QV30 level accuracy at the single molecule level. So the end result is a highly accurate, maybe Sanger level accuracy of a 15 kb or greater insert. And I just want to compare this with what you might think of as traditional long-read sequencing. So in traditional long-read sequencing, you have a single pass on a long molecule, could be very long, but this is single pass accuracy. So this might be, let's say, 86% accurate. And then the way that this works is that you take multiple molecules and you align them together bioinformatically and then resolve the errors across multiple molecules to reach high consensus accuracy. So this is something that you absolutely can still do with the Sequel II system. There are particular applications where we would recommend this, and I'll go into that. But really, what's new and unique and really exciting for us is this high accuracy long-read sequencing. Also, just a couple weeks ago, we have our latest generation of chemistry for the Sequel II system. So this is our 2.0 chemistry. So I want to show some results for that. So this is a read length distribution for one of those 15 kb HiFi libraries. This is a human data set, HG2. So you can see that this is kind of a typical read length distribution. The total raw data that we were able to get from this cell was about 450 gigabases, so that's about 5 million reads. And the read length N50 is 185 kb, with reads extending out over 300 kb in length. I have a different type of graph I want to show you. So you're probably used to seeing read length distributions from us. This is an accuracy distribution. So across the x-axis is the Q score. So at the color change is 20, then 30, 40, 50, out and further. And then on the y-axis is the number of reads that meet this quality. So for this particular data set, after processing it for HiFi, we had a mean accuracy of Q34. But I just want to point out that that means that half of all reads are actually higher accuracy than Q34, as you can see in the distribution graph. I'll also mention that we have this data freely available for download, so if anybody's interested in looking at it, let us know. The other thing that we're able to do with the new 2.0 chemistry is we're really pushing the limits on insert size for HiFi libraries. So here are two libraries. We have a 20 kb insert and a 24 kb insert. And because the x-axis is kind of small, I've denoted with the hash line where Q30 is. So you can see that the bulk of the reads are at or above Q30, even for these very large insert libraries. And just going back to that continuous long-read sequencing, so away from HiFi now, here's a read length distribution for a 35 kb insert library. So you can see that we've got about 180 gigabase of data for the CLR mode of sequencing, and half of all of the bases are in reads greater than 50 kb, with a long tail out greater than 175 kb. So this is looking very good as well. So I'm going to switch to applications now that we're up to speed on performance. So I'll talk about each of these in detail, some a little bit more than others, but the ones that are of most relevance for this audience is going to be in the area of human whole genome sequencing. We have three primary applications there. So I'm going to talk about the first two, which are both variant detection applications. So these are kind of resequencing and alignment type applications. And I'll differentiate them for you. So the first one is what I'll call high-throughput structural variant detection. And this is one of those applications that I mentioned that makes use of the CLR, continuous long-read sequencing. And so this application is useful for detecting larger types of variants, like structural variants or copy number variants. Because of the high-throughput nature of this application, this is really well suited for population studies. So the experimental design here is that you're able to multiplex 2 samples per SMRT cell. And so what that gets you is a cost per sample of $670. So it's very low cost per sample, and you're able to do about 1,000 samples per year per system. So I want to compare that with the other structural or the other variant detection application that we have, which is comprehensive variant detection. And so this is using that HiFi or high accuracy long read sequencing to detect variants. So you can still detect structural variants and copy number variants, but here, because of that high base level accuracy, you're also able to detect single nucleotide variants and indels as well. So what we're finding is that people in the area of disease research are finding this application very useful, maybe especially in rare and undiagnosed disease, where you've tried other types of technologies and you haven't found an answer. This application is great for that. The experimental design here is one library spread across 2 SMRT cells, so 2 SMRT cells per sample, and a cost per sample of about $2,600. And then the throughput here is relatively lower, it would be about 100 samples per year per system. So I'm going to stick on the HiFi variant detection, the second one that I described earlier. If this is of interest, I would direct you to this paper that my colleagues, as well as a large number of external collaborators, published just in August. Here in the paper, we looked at the precision and recall of variant detection with HiFi reads. These are actually Venn diagrams, but they overlap really, really well, so they mostly just look like circles. But this is our comparison of our HiFi data set compared with the genome in a bottle benchmark. And so you can see for SNPs, indels, deletions, and insertions, we have very, very high precision and recall with this data set. The other thing that's great about the HiFi reads is that we get much better mapping in the human genome. So here's an example of the STRC gene in chromosome 15, where you can see in the top panel there are a number of exons where short reads just don't map. And then in the lower panel are the PacBio HiFi reads. And you can see that they map just fine across the whole gene, and we're actually able to call SNPs that would not be possible to call otherwise. And maybe this is just one gene that's cherry-picked, but there's actually something like 200 medically relevant genes that we know of where it is challenging to map all of the exons with short reads. And so in this paper, we looked at all 193 of these, and we found that for about 75% of them, the HiFi data were able to map to 100% of these problem exons. And for the majority of the others, we were able to at least get some of the exons, with only just some in that bottom red panel where we were not able to map. I would point out that this was with 13 kilobase inserts, so as we push that HiFi read length longer, we expect to get better mapping in some of these most challenging genes. So I'm going to go back, just to kind of reset. We're going to go back to the high throughput variant detection. So just to refresh, this is using the continuous long read sequencing. And this is the one that's high throughput, 1,000 samples per year per system for relatively low cost. And this is really ideal for larger size cohorts, so population studies in the hundreds or even thousands of samples. So now that we have this really high throughput, thanks to the Sequel II system, we're able to address cohort sizes, and we can detect variants in the population down to 1% frequency or even less. And typically what we recommend for experimental design here is that, let's say you're working on a cohort that's tens of thousands or even hundreds of thousands of samples. What we see people doing here is using PacBio to detect structural variants in a smaller subset of those, and then combining that with short read sequencing to call variants or genotype variants in a larger population. So it's highly complementary in that fashion with data that's typically already available. And indeed, we're seeing really good adoption of the variant detection application. So here's just a couple press releases. One from September, which is the SolveRD initiative in Europe, and the Radboud University in Nijmegen in the Netherlands. And they're using PacBio to call variants in a cohort size of 500 whole human genomes. And then just yesterday, we had an announcement from Discovery Life Sciences at the Hudson Alpha Institute that they are setting up a center of excellence for PacBio sequencing for a number of different applications, but most primarily to detect variants in large human cohorts. And Sean will be talking a little bit more about that shortly. So the last human whole genome sequencing application is whole genome sequencing for de novo assembly of reference genomes. I think this is probably what PacBio is most well known for, so I won't talk about this for too long. Other than I want to mention what's new. And what's new is that we're now recommending that you use HiFi data for de novo assembly of human genomes. The reason being is that you're able to achieve comparable contiguity as you do with CLR, but at much higher base level accuracy. So if you just compare a HiFi human genome assembly with a PacBio CLR human genome assembly, we have 6 times higher accuracy. And if you compare that, for example, with an Oxford nanopore genome that has been error corrected with Illumina, we actually have 80 times higher base level accuracy than those assemblies. So this really is the best way to generate highly accurate human genome assemblies that's available today. There's another benefit of HiFi data, which I don't think should be overlooked, and that is computational requirements. So with the HiFi data, we cut analysis time in half. So with CLR, it's about 60 hours, and with HiFi, it's about 30 hours. And you will notice that file sizes as well. The input file size is about 1/6 of the size as with CLR. So if you're talking about scalable de novo genome assembly, HiFi is a much better solution on the computation side as well. And there are a number of people generating PacBio reference genomes out there, so we're really excited to be a part of the new era of ethnic-specific reference genomes. There are nearly 40 of them that have been published to date, represented in this map here. Most recently, I'll just mention a couple weeks ago, there was a preprint that came out for the Tibetan genome assembly, which was done with PacBio data and has some really interesting findings about adaptation to high elevation. As well, there's a recently funded project by the NHGRI to generate the human pangenome. So this is to sequence and de novo assemble 350 ethnic-specific reference genomes And in the initial phase, they plan to generate both PacBio HiFi as well as CLR data. And we're really happy at PacBio to be a part of this project, along with a number of other sequencing companies. So I'm going to just touch briefly on a couple other applications. So in the area of RNA sequencing, full-length RNA sequencing with our IsoSeq method, we're really happy to introduce a couple months ago the IsoSeq Express kit. The name Express implies that it has significantly speeded up the library prep time for IsoSeq. So you can now go from total RNA to a sequencing library in about a day. We've also significantly reduced the input requirements, eliminated the need for size selection in the protocol that's associated with this, and we've reduced the cost of template prep by 1/3 for IsoSeq. And what that translates to on the Sequel II system is that we can now reach saturation in the human transcriptome in 1 SMRT cell per sample. So that's a comprehensive human transcriptome in 1 SMRT cell. We also have some improvements on the software side. I just want to mention IsoPhase, which is a tool that's available for phasing RNA data. And basically what that means is if you want to explore allele-specific expression and look at alternative transcription, we have a tool available for that. We also have a very active group of collaborators and customers in the area of IsoSeq who are developing a number of interesting methods. So this is DartSeq. It was just published recently. This is a really creative method for using IsoSeq to detect 6-methyl-A in messenger RNA. So if that's something of interest, you can check out this paper. So like I mentioned, you can do a human transcriptome in 1 SMRT cell. So on the affordability side, that means that it is about $1,300 per sample for human transcriptome. And back to the chemistry version 2.0 launch, here's what it looks like with an IsoSeq library. So you can see that in total, we got about 0.5 terabase of data in 1 SMRT cell, human transcriptome. And after processing and mapping, we've got about 210,000 mapped unique isoforms. So there's probably some interesting biology that can be done there. If you'd like to learn more about IsoSeq, I would encourage you to visit my colleague Liz's poster. It's poster 1455, and it's in the session this afternoon. I'm not talking about it here, but we are also working on single-cell IsoSeq. So both internally and then with collaborators and external customers have been working in this area. So if that's something of interest, then I would encourage you to attend Hagen Tilner's talk this evening. He'll be talking about single-cell IsoSeq. As well, we have a collab talk on Friday. So it's Friday at 10 o'clock in Theater 3 on the exhibit floor. Liz will be going over IsoSeq in a lot more detail. And as well, my colleague Aaron Wenger will be talking about structural variant detection. So I'd encourage you to attend that for a deeper dive. Targeted sequencing. So of course, with really high throughput, you can pretty much do any targeted sequencing experiment in 1 SMRT cell. We're multiplexing to a very high level for amplicons, and it's now just a couple dollars per sample, essentially, for amplicon sequencing. We also launched a new method recently, which is no-amp targeted sequencing. And this leverages the CRISPR-Cas9 system to enrich areas of the genome that are difficult to sequence otherwise. So you can think about repeat expansions, for example. So if you're interested in learning more about the no-amp method, I would highly encourage you to come to our booth. Here's our lovely booth staff. We're very happy to chat with anybody, so come on by. And we do have a poster available in the booth that covers the no-amp method. So lastly, I'm just going to mention metagenomics if anybody is doing microbiome work. As well, pretty much any metagenomics experiment can be done in 1 SMRT cell for a very reasonable cost, whether it's 16S up to full metagenomic assembly. If you're interested in metagenomics, again, just come and see us at our booth. And lastly, in addition to our workshop here, we have a number of customer talks across the whole program of ASHG as well as posters. So if you're interested in seeing some more of that, we have flyers available at the door, so please grab one on your way out, and that has the full list of talks here. And lastly, I promise, is that we are happy to announce the 2019 Human Genetics Smart Grant Program. So this is open to anyone, and you're welcome to propose any project either in the area of human genomics or transcriptomics. And the award of the grant is 12 SMRT cells on the Sequel II system. So if you were take away anything, you would know that there's a lot you can do just with one SMRT cell. So this award is 12. So think big for this one. And we're taking applications until November 22nd. And we're really happy to be doing this in collaboration with Hudson Alpha as well.